Hello everyone, my name is Niveditha Nerlakanti and I'm a PhD student in Tumor Biology Department at Moffitt Cancer Center. Here at the AACR 2022, I'm pleased to present my study that investigates how epigenetic upregulation of neuropilin-1 promotes osteosarcoma progression and metastasis. Osteosarcoma is the most common bone cancer and it frequently metastases to lung tissue. And the prognosis for these patients is grim, with 5-year overall survival rates as low as 30%. There has been little change in the treatment paradigm for this disease from past 4 decades, and new targeted therapies are an urgent and unmet clinical need. We have previously identified HDAC inhibitor panopinostat, which is FDA approved for multiple myeloma disease, to be most effective in limiting osteosarcoma growth in vitro and in vivo. Due to broad spectrum nature of panopinostat, inhibition of HDACs has noted toxicities in humans. To address this, using siRNA approaches, we identified that HDAC1 and HDAC2 are important for osteosarcoma survival. And this is further supported by our data showing the efficacy of HDAC1 and 2 specific inhibitor romidepsin. Romidepsin, like panopinostat, also has noted toxicities. We therefore decided to determine the potential mechanisms through which HDAC1 and 2 mediate their effects. Subsequently, we performed microarray analysis on romidepsin-treated osteosarcoma cells to identify key up and down regulated targets of interest. We identified neuropilin-1, a membrane-bound co-receptor for VEGF, to be highly downregulated in response to romidepsin. Interestingly, NRP1 knockdown by shRNA in osteosarcoma cells led to significantly reduced cell proliferation and also cell migration. Further, we performed phosphokinase array, which revealed increased phosphorylation in MAP kinase pathway proteins in NRP1 knockdown cells. Based on this data, we conclude that HDAC1 and 2 contributes to osteosarcoma growth potentially via the upregulation of NRP1, a therapeutically actionable target. If you have any further questions about my research, please visit my poster at AACR 2022.